Okay, so first of all, just a brief disclaimer. So the information presented is not the official opinion of Michigan State University or the University of New England. Um, I was a professor at Michigan State University for 30 years, now retired. Lele, my co-host, is a professor at the University of New England, Biddeford, Maine. And in the School of Biological Sciences, I was in the Department of Biochemistry and Molecular Biology. So this is a Life on Earth, Origin of Life webcast. The topic here is the genetic code, and um, it's hard to be absolutely certain about how the genetic code arose. So what I'm giving you is a working model for how the genetic code evolved. My co-host is Lele, and that's me. So first, some subliminal messages. So this is a working model. So what we've told you about transfer RNA is a theorem. It's a proven model. This is a model with a different status. I'm going to say that tRNAomes, all of the tRNAs from an organism, aminoacyl tRNA synthetase enzymes, this group of enzymes for an organism, and the genetic code are all co-evolved. Other things are co-evolved with this, but you have to somehow integrate all of this information, all the tRNAs, how the tRNAs are recognized and charged, and then how the genetic code is output to understand these various processes. They all need to be integrated. I think what I'm presenting overall is the greatest story in 4 billion years. So this is a big deal. There are bottom-up and top-down approaches to origin of life. So a bottom-up approach would be making life or partly making life in a test tube, which remains a challenge. And a top-down approach, we take a top-down approach partly because we're molecular biologists. So basically, we're trying to infer how the origin of life started through existing biology, in this case, the genetic code, how the genetic code was founded. So what I'm going to say is that the genetic code is non-overlapping and it's also degenerate. What I mean by degenerate is that there are too many codons, too many anticodons encoding too few amino acids. But degeneracy results from wobbling. So the wobble position in the anticodon is tRNA 34, but tRNA 34 and 36 used to be wobble positions, and wobbling was suppressed at tRNA 36. And this is how we're going to say the genetic code evolved. So transfer RNA, have to know something about that. tRNAomes, all of the tRNAs of an organism. Aminoacyl tRNA synthetase enzymes, there are about 20 of these, one for each amino acid in most organisms. So the genetic code evolved, we're going to say, by this pathway. So initially, the genetic code was a nucleotide-dependent system for synthesizing polyglycine. So glycine's the simplest amino acid. We're saying it was the first amino acid. And after that, glycine, alanine, aspartic acid, and valine, GADV world. So I'll explain. Other people have said the same thing, that GADV world came before the world of protein synthesis that we understand today. Then you go to glycine, alanine, aspartic acid, valine, leucine, serine, glutamate, and arginine. So leucine, serine, and arginine all become 6 codon sectors. We're going to explain how we think that occurred. Glutamate is a similar amino acid to aspartic acid. And so we're going to say that glutamate and aspartic acid sort of hitchhiked into the code almost together, but with aspartic acid coming first and then diversifying to include glutamate. And then you add some other amino acids, and then finally you fill the code by adding aromatic amino acids, phenylalanine, tyrosine, and tryptophan. These are those and cysteine, these are aromatic amino acids. Cysteine is a late addition to the code, although cysteine probably hitchhiked in on serine, as I'll try to explain. Methionine is also a late addition to the code. Methionine invades an isoleucine 4 codon sector, I'm going to say. So anyway, a rational explanation can be given for the entry of every single amino acid into the code. So lots of people complain that there's a chicken and egg problem in evolution of the genetic code, because how can the code know when it starts out evolving that it's going to have nucleotide sequence-dependent synthesis of proteins? And the answer to that is that the synthesis of polyglycine was strongly selected, and that's a nucleotide-dependent reaction. The synthesis of glycine, alanine, aspartic acid, and valine polymers, that was strongly selected. So the system is strongly selected from the very beginning. It simply learns new capacities as you move across the timeline of its development. So what I'm going to say, the genetic code evolved around the tRNA anticodon, and position 34, 35, and 36 form the anticodon. 34 is the wobble position. This is a Watson-Crick and a Watson-Crick position. So 4 possibilities here that can interact with A, G, C, and U in messenger RNA, and this one can interact with A, G, C, and U with messenger RNA. But this one, there are only 2 possibilities, purine versus pyrimidine. So only 2 possibilities here, 4 and 4, maximum complexity of 32 assignments. But we ended up with 20 amino acids plus stops. In a textbook, it'll tell you that the complexity is 4 times 4 times 4 is 64. That's misleading. That ignores where code degeneracy comes from. But initially, I'm going to say that this was a wobble position and that was a wobble position, and then wobbling was suppressed at this position. But before you suppress wobbling at 36, then you have 2 possibilities here and 4 here. This is always the easiest position to read, or 2 times 2 possibilities here and 4 here. So you hang up at 8 amino acids as you're evolving the code. So you hit a bottleneck at 8 amino acids, and then you have to suppress wobbling here in order to advance. And part of suppressing wobbling at the 36 position is this heavily modified base, which is part of the reading of this position. And also there's a conformational change in the ribosome small subunit that allows Watson-Crick geometry here, enforces it here, and regulates wobbling here. So the genetic code evolved around the tRNA anticodon 34, 35, and 36 in tRNA numbering. initially 2 wobble positions, 36 and 34, evolved to 1 wobble position, that's 34. You can't give up wobbling at 34 to add more amino acids to the code without significant difficulties. That would be a lethal mutation at this point. And suppression of wobbling at tRNA 36, that involves 30S ribosome closing mechanism that enforces Watson-Crick geometry here and here and regulates wobbling there, and also the heavy modification of tRNA 37 to space here. So 2 ways that wobbling at position 36 is suppressed. So what does the genetic code look like? I'm going to say that the genetic code is highly ordered. It should be thought of at its inception as a 32 assignment code, not a 64 assignment code, as I explained. I've already given some background on how these positions were interpreted. This is a highly ordered system. These in yellow, leucine, isoleucine, methionine, valine, those are hydrophobic amino acids. And these are all 1A aminoacyl tRNA synthetase charging enzymes. So lots of evolution in columns. That's evolution in column 1. Evolution in column 2, you have serine and threonine are closely related. These are all neutral amino acids. These are all 2A enzymes. I'm going to say that alanine was initially a 2A enzyme that was replaced before LUCA, the last universal common cellular ancestor, with a 2D enzyme. And here you have this lovely stripe pattern: glutamate, aspartate, lysine, asparagine, glutamine, histidine, orange, green, orange, green, orange, green. Column 3 is the most innovated column in the code, and there are reasons for that. I'll give explanations for that, but lots of evolution in the column. Orange, 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 which are all 1B enzymes, and asparagine and histidine. This used to be a 2A enzyme. It evolved to a 2B enzyme to discriminate it from his2A, his-RS2A, and then asparagine 2B evolved from asp2B. And here you have arginine and cysteine, both 1A enzymes. Serine jumps in the code, and I'll have to explain that. I think serine used to occupy this entire sector, and it jumped there. I think probably cysteine was part of serine jumping, and then cysteine lands in row 1. Row 1 is late to be filled in the code because when this is a wobble position, 36A is not allowed. 36A is not allowed. You can't fill row 1. You can only fill row 1 when you lock down 36. And you lock that down by this modification, which comes late in code evolution, and also when you get this conformational change in the ribosome. As you can see here, no A is allowed in the anticodon in the 34 position. That's 34 down there. A is not allowed in archaea. In other organisms, A is really only allowed when it's modified, for instance, modified to inosine. Okay, so what am I forgetting to say? So just a little bit about this figure. This is the codon, first position. second position, third position, that's the messenger RNA. This is the anticodon, 34 and 34, but U and C are essentially equivalent. And G satisfies the need here because G in the 34 position interacts with U as a wobble base and with C as a Watson-Crick base. A isn't needed and it is never used in archaea, or close to never. There may be exceptions to that that I don't know about. This anticodon was killed here. That's by methionine and fading a 4 codon isoleucine sector. Serine jumps. I'm going to say it jumps from here to there. Threonine is a related amino acid to serine. So this is somewhat of an orderly event. And then I think this was probably a cysteine. Serine can be converted to cysteine attached to a tRNA, and then cysteine can occupy this position and serine can occupy this position. I think that's how serine got there. Glycine, I'm going to say, initially filled the entire code except for perhaps row 1, which was mostly stops. And glycine ends up here. This is the most favored position in the code. It's the most favored position because it's 35C, 36C. So this is 35 position here, always a C in row 4. And that's row 4, that's column 4. Row 4, C is 36 position all the way across. So one thing that this indicates is that glycine, aspartic acid, alanine, and valine, GADV, were the first 4 amino acids into the code. Anyway, I'll continue to try to make sense out of this figure here. It takes a little study. I mean, it took a little study for us, so it might take a little study for you as well. Okay, so there's much evolution in code columns. That's the 35 position. Code columns. Column 1, that's always A. Column 2, that's always G. Column 3, that's always U. Column 4, that's always C. The 35 base is the most important base in evolution of the code. The code appears to fill up by rows, row 4, and then 2, and then 3, and then 1. Row 4, that's 36C. This is rows. And then 2, that's always G, 36G, all the way across. And then 3, that's U. And finally, row 1. Row 1 is disfavored because that's 36A, and that's a wobble position initially, at which wobbling is eventually suppressed. Columns 1, 2, and 4 sectored mostly on 35 and 36, 35 and 36. So sectored mostly by rows and by the 35 position. So column 1, 2, and 4 sectored that way. That's why you get so much evolution here and why you get such large sectors. That's a 4 codon sector there. And in column 3, you get the most innovated column, and that's most innovated because you were sectoring on the 34 wobble position and 35. Initially, there's 1 wobble position can be used. That's either 36 or 34. Columns 1, 2, and 4 sectored on 35 and 36. Column 3 sectored on 34 and 35. And there's no A in a wobble position. And I keep saying this, but you can't forget it, because if you forget it, you can't understand degeneracy or how the code sectored. That's important. And in archaea, never A, at least so far as I'm aware, never A. in the 34 position. There may be exceptions, but I don't know about them or how to explain them. Okay, so initially we have polyglycine world. So what I'm going to say is that pretty much the whole code encodes glycine. And so how do you understand that? So basically, you don't have any proteins yet, but you can have primitive catalysts such as ribozymes. So PC in this case is not politically correct. It's primitive catalyst, okay? And those can be ribozymes. Think of those like RNase P, that is to say ribozymes with other features. And the other thing, there's no 34A and no 36A because there's no wobble A. So these are all stops across row 1, no 36A. And so how do you think about this? So the way you think about this is that the anticodon within the anticodon loop is the easiest part of tRNA to modify, because if you modify other parts, you affect stems and then you need compensating mutations. But it's easy to imagine rapid innovation in the anticodon to many different sequences. And this starts to give you evolution in columns. You're starting to figure out now how to get the column 4 occupied. And then you go to glycine, alanine, aspartic acid, and valine. So G, A, D, V in the one letter code. And now we've explained evolution in columns 1, 2, 3, and 4. It follows the entry of these first 4 amino acids, all of which are going to land on row 4 of the code. And all of these are very simple amino acids. Valine is a simple amino acid. Alanine is a simple amino acid. Aspartic acid is a simple amino acid. And glycine is the most simple amino acid, and it's the first one in. Okay, so now we're approaching our 8 amino acid bottleneck. And to explain that, so you have an 8 amino acid bottleneck as long as both position 36 and 34 are wobble positions. And in order to go beyond 8 amino acids, you probably have to lock down position 36 and make it into a Watson-Crick instead of a wobble position. And I'm also trying to partly explain how leucine, serine, and arginine become 6 codon sectors in the code. I'm also trying to explain this orange-green, orange-green, orange-green pattern in column 3 of the code that leads column 3 to encode the most amino acids and become the most innovated sector of the code. Valine, alanine, aspartic acid, and glycine are all going to land on row 4. Row 1 still can't be occupied because you haven't locked down the 36 position yet. So these are stops. They may not be efficient stops, but they're stops in the early evolution of the code. Then I'm going to say that leucine, serine, and arginine invade down. And these amino acids are perfectly happy because they're in the best row of the code. So they're giving up space in the code, but they're occupying the best row, which is row 4, that's 36C, and the best column, that's 35C. That's why glycine ends up in this 4 codon sector. And this is also a partial explanation why leucine, serine, and arginine become 6 codon sectors. So we're still hung up at 8 amino acids, and we can't break the bottleneck till we suppress wobbling at the 36 position. Because until then, you can't utilize row 1 efficiently. But this does set up the possibility for serine to jump from here to there, which it's going to do. Oh, there's serine jump. So now I've messed up my order of events here because by rights, I shouldn't be able to fill these positions yet because this is still a stop codon. But the order of events isn't as important as getting the concept right, I think. So the concept, serine is going to end up here and here. Leucine is going to end up there. Phenylalanine there. Serine is going to jump from here to there, but this may be serine that's modified to cysteine. So serine can be modified to cysteine on a tRNA. That happens in some organisms. And then cysteine could jump here and subsequently move up to its eventual resting place, which is there. And then this reverts to become part of a serine 6 codon sector. You still have this lovely stripe pattern of glutamate aspartate, glutamate aspartate, glutamate aspartate here. And that's sectoring on the 34 and 35 positions. And you can see that this is 34 and 35, the A and B rows. Here you're sectoring A, B, 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 like that. So now we're going to break our bottleneck. We're going to start to suppress wobbling at the 36 position. And I haven't done it completely. These end up to be the positions of stop codons. So it makes sense that it's harder to suppress wobbling in these positions, perhaps, than in these positions. Serine ends up there. Serine jumps there. Threonine replaces serine. Threonine is a related amino acid to serine. Proline enters the code there. Leucine is getting close to its final form. Isoleucine is a 4 codon sector and utilizes 3 anticodons, but it hasn't been invaded yet by methionine. And here we have arginine. Eventually, cysteine is going to occupy that position, and tyrosine, tryptophan here, phenylalanine there. And then the code will be complete. I think I just finished the code. So when you suppress wobbling at this position, row 1 can be completely occupied. The last amino acids in are the aromatic ones, tryptophan, tyrosine, and phenylalanine. I'm going to tell a little story about that in a minute. And cysteine ends up here, but we think that cysteine may have hitchhiked in on serine and been part of the mechanism by which serine jumped from column 2 to column 4 in the code. Valine, alanine, aspartic acid, and glycine, G, A, D, V, land on row 4, which is favored because that's 36C. 36C is favored, and 35C is favored. So this is a favored column in the code. This is a favored row. You end up with aspartic acid, asparagine, and histidine. And I'm going to say that this was initially a 2A enzyme, and then it deviated from his2A to become asp2B, and then later asparagine 2B evolved from asp2B. there are systems in place that will convert an asparagine tRNA linked to an aspartic acid into asparagine. So that's tRNA linked chemistry. There's tRNA linked chemistry with tRNA serine being converted to tRNA cysteine, or a tRNA cysteine loading serine and then being altered to cysteine. And the same thing with glutamine and glutamate, which are related amino acids. So you can link glutamate to a tRNA glin and then convert the glutamate to glutamine enzymatically by a second enzyme linked reaction. Okay, so what I'm going to say is that probably initially phenylalanine occupied these sectors and probably as a type 1C enzyme. So this used to be phenylalanine 1C and then got replaced by phenylalanine 2C, but that occurred before LUCA, so there's no sequence record of that event. Oh, and here we have methionine. So you have to spend a little attention to the one codon sectors. So methionine uses the CAU anticodon to recognize AUG. Isoleucine also utilizes CAU in archaea, but it utilizes C-modified tagmatidine, and the modified C recognizes AUA, but not AUG. And so that's how isoleucine and methionine can share a single anticodon through tRNA modifications in the wobble position. And also, this is how methionine invaded an isoleucine 4-codon sector. If I back up one, now you have a 4-codon sector encoding isoleucine, and you have used these 3 anticodons. And then when methionine invades, it kills this anticodon, and then it and isoleucine have to share CAU, but isoleucine modifies the C tagmatidine so that this can be recognized AUA and AUC or AU or AUU for isoleucine. So did all that make sense? I'm hoping it's starting to. It actually requires some effort to break this information down. And I think what I've done is given a rational explanation for the placement of every amino acid in the genetic code. And that's the first time that that's ever been done. There are other models for genetic code evolution, but they're not as detailed and they don't make as many predictions as this one. Okay, so now we're back to this figure, and that's the wobble position and the 35 position and 36. This is 34 down here. And I hope I've begun to explain why there's so much order in the genetic code and why row 4 fills early and why the first amino acids, G, A, D, and V, enter early, and why glycine, which initially fills the whole code, ends up in row 4, which is 35C. So this position wants to be C, that wants to be C. So this is the most favored anticodon in the code. There's nothing random about the placement of amino acids in the code. There's very little chaos in evolution of the code. And I think that begins to make sense. And it begins to make sense the harder you study it, the better sense it makes. It's hard probably at first hearing to get all the details. I'm not sure. Maybe I'm doing a perfect job of presentation. I'm not sure about that. Okay, so what I've presented is a working model. I don't know exactly how this process went down. This is such a good working model. I think it's probably mostly right, but that remains to be seen. It makes many predictions, so it makes many, generates lots of ideas for both simulation and for other approaches to try to confirm or improve the model. It's completely rational. There's a rational explanation for 1 codon sectors, 2 codon sectors, 3 codon sectors. There's only one of those for isoleucine, 4 codon sectors, and 6 codon sectors in the code. Makes lots of predictions. And it explains, I think, why you get evolution in columns. This is evolution around the 35 position of tRNA, which is the most important for evolution of the code. Also, evolution by rows, that's the 36th position. This starts out being a wobble position. You suppress wobbling. It appears that the code fills in row 4, followed by row 2. 4 is C, 2 is G, 3 is U, and finally 1 is A. To utilize 36A, you need to suppress wobbling at the 36th position. There's a stripe pattern in column 3. That's our green, orange, green, orange, green, orange pattern. And that has to do with selection of 34 as a wobble position. So that's what's different about column 3 and columns 1, 2, and 4. Leucine, serine, and arginine end up in 6 codon boxes. And I hope I explained how that occurred or how it might have occurred. Serine jumps from column 2 to column 4, and I think this is part of cysteine hitchhiking in to the genetic code on serine. And then cysteine ends up in row 1, column 4, as a result of this jump. Cysteine, it's important to include cysteine in even in an early developing genetic code because cysteine is so important for retaining metals. And retaining metals is a big part of what proteins do. I've explained code degeneracy. So code degeneracy comes from degeneracy at the 34 position, but also initial degeneracy at the 36 position. And why glycine is the first amino acid and in the most favored anticodon position. Glycine ends up with the best anticodon. It's the first in. It occupies the entire code and then it retreats to the best position. So glycine placement in the code is not random. There's no A in an anticodon wobble position. I don't actually know all the reasons why this is true. And if anybody out there knows that, they should send me an email. Why no A in an anticodon wobble position? Well, G suffices because G recognizes U as a wobble base and C as a Watson-Crick base. But universally in archaea, so far as I know, there's no tRNA34A. It's not allowed. And why row 1 fills last? That's because when 36 is a wobble position, A is not allowed in a wobble position, just as A is not allowed in the 34 wobble position. Row 4 is favored. That's third anticodon position C, tRNA36C, and column 4 is favored. That's second anticodon position, tRNA35C. So having C in the 35 and 36 position is preferred over other bases in evolution of the code. So with the code, you get lots of things. You get biological diversity and complexity without nucleotide-dependent synthesis of proteins and specification of proteins sequences. You can't have biological complexity. It's just not possible. But with the code, this becomes possible. So that's why you get a code. And so what are scientific working models for? Good question. Okay, why bother to think of such things? So one reason is to seek rational explanations that otherwise can't have a rational explanation. Okay, so you can either decide that evolution of the genetic code is an act of God or happened all by itself by a random process. That's obviously not true. Part of this working model gives a rational explanation for evolution of the genetic code. So you can throw up your hands and just give up, or you can try for a rational explanation. I think I've given the basis to plan for many experiments. For instance, why is A not allowed in a wobble position? I think that's a good experiment for people to do, or can also be done computationally. You can plan tests, so many sequence analyses should be informed by the information I've provided. You can plan simulations, so there are lots of people trying to simulate the evolution of the genetic code. Most of them use random additions of amino acids, which are meaningless. And to see connections. So there are connections between tRNA evolution, tRNAome evolution. tRNAome is all the tRNAs of an organism. Aminoacyl tRNA synthetase evolution, there are up to 20 of these for a species, and genetic code evolution. These are all linked processes. And you can see that just from what I've presented, but you could also attack this using machine learning and artificial intelligence to see if you could gain further insights. So should you be skeptical? So it's fine to be skeptical, but be flexible. Don't be so skeptical that you don't utilize all available knowledge and information and ideas. So, for instance, climate change deniers think that they're skeptics. They're not skeptics, they're just deniers. They're not thinking about the available information. Is a working model explanatory? That's a good test for it. Is it useful? And I think the model I've just given you is really useful. And that's a bunch of my papers and some of Leigh's papers on the subject. We've been working on this for a few years now, I think. Isn't that right, Leigh? Several years. And that's the end of my presentation. So why don't we stay here, because I might need my figures to answer questions.
Sure. I have several questions. You present a multi-step model on the evolution of the genetic code, and you also divide that into several distinct steps. So I have some specific questions regarding some of these amino acids. The first question is, how does methionine enter the code?
Okay, so yeah, I thought this would be necessary. So what I'm going to say is that methionine, so you have to explain methionine and you have to explain tryptophan, because those are in one codon boxes. And you shouldn't have any one codon boxes.
Okay.
as I explained. But I'm going to say that methionine became a one codon box by invading a 4 codon isoleucine sector and killing this tRNA. And then you use tRNA modifications to discriminate CAU from methionine, which is largely unmodified or lightly modified, and isoleucine, in which this is modified heavily to agmatidine. And I guess agmatidine looks a little bit like G, so that you can recognize these two anticodons for isoleucine. So if we back up one, we have isoleucine as a 4-codon sector, happily using 3 anticodons, G, U, and C. And then methionine invades. Methionine has to invade because it's the initiation codon, so it's needed for initiation and elongation. So you can't have regulated starts without the invasion of methionine or some other mechanism to do that. So methionine invades, it kills this anticodon, and then isoleucine and methionine have to share CAU, but isoleucine modifies C to agmatidine, which looks like guanine, I'm going to say, and methionine uses a lightly or unmodified CAU. And there are 2 methionine tRNAs, one for initiation, one for elongation, so you can have a regulated initiation mechanism. So that's methionine. That's a pretty good story, I think. It took me a long time to figure that out. So if you want to figure this out by reading lots of papers, you should start with my papers and then move on to some of the papers I reference.
Okay.
That's my suggestion.
My next question is, how does cysteine enter the code?
Cysteine. Okay, so yeah, that's a long story. So what I'm going to say is that this serine box may have jumped over to here actually as a cysteine box. So serine can be converted on a tRNA into cysteine. And there's still an organism or two that will do this. So if this is actually cysteine hitchhiking on serine by a 2-enzyme mechanism, it could end up over here, and then cysteine could end up here, and this could revert to a serine box. Serine ends up here and there. So serine jumping in evolution of the code is part of the only chaos that occurs in code evolution, I'm going to say, or the only remaining chaos. That mostly everything is an ordered process, but serine jumping seems like a little bit of chaos, but it's only a little bit of chaos. It's not a lot of chaos. And I think it's linked to cysteine ending up in this box here. So from here, cysteine jumps up there. It's easy to jump there. It only requires a single base change in the tRNA. And then this can revert to serine. This can be here, serine can be replaced by threonine.
Okay. So my next question has to do with stop codon. So how do you think stop codon evolved?
Okay, so stop codons. So one thing I think I failed to mention is that stop codons don't have their own tRNA. So there's no tRNA here, there, or there. And stop codons are recognized in messenger RNA. So this is messenger RNA here. So a stop codon is bound by a protein release factor that releases the messenger RNA from active translation. And there are 3 of those, that one, and that one. And those are systematic absences in tRNAs. And so what I'm going to say is that part of this and this ending up as stop codons has to do with the late suppression of wobbling at the 36th position. And that this is already a kind of a stop codon, and then you evolve the ability to make protein release factors late in code evolution. And this tRNA, that tRNA, and that tRNA end up being stop codons. Was that an okay explanation, or am I missing something?
Sure, of course. Of course.
Okay.
So the next question is on phenylalanine. So how do you think phenylalanine enters the code?
Okay, so phenylalanine ends up here, and it ends up being a 2C aminoacyl tRNA synthetase. So I'm going to say that phenylalanine wandered across row 1, and then it landed here initially as a 1C enzyme, but now you have tyrosine aminoacyl tRNA synthetase 1C. And so to discriminate tyrosine from phenylalanine is difficult for an enzyme because it just differs by a hydroxyl group. So part of that before LUCA, the last universal common cellular ancestor, you replace phenylalanine 1C with phenylalanine 2C, just as here you replace an alanine LRS 2A with a 2D enzyme for LUCA. Because it happened before LUCA, there's now no sequence record in existence, so far as I know, of those events. That's a model.
Okay. So how about asparagine? How does asparagine enter the code?
Okay, so there's actually a good sequence record of asparagine entering the code. So in a few organisms, you can add aspartic acid to an asparagine tRNA and then convert it in place to an asparagine tRNA by a second enzyme. And the second mechanism for the same mechanism for glutamate and glutamine. So to add glutamine initially, you have a glutamine tRNA with bound glutamate added by a GluRS, and then it gets converted by a second enzyme to glutamine attached to the tRNA. There's a lot of tRNA linked enzyme chemistry, and that's a big part of the evolution of the code. That's also how I'm saying that cysteine entered the code. Initially, this was tRNA with serine bound, and a second enzyme converted it to cysteine, and then cysteine ended up on as a late final entrant into row 1, but I think cysteine was added into the code by tRNA linked chemistry as an earlier event.
That brings me to the next question. Is RNA-linked metabolism co-evolve with the code?
So yes, I'm going to say that that RNA-linked chemistry is a very early event in evolution of tRNAs and in evolution of proteins and protein coding. And it's part of how cysteine entered the code. It probably has something to do with serine jumping, as I tried to explain, has to do with aspartic acid and asparagine, these two enzyme systems, and glutamate converted to glutamine. That may be part of how some of these amino acids also entered the code, because when you have this whole column encoding valine, valine can be converted to leucine and isoleucine in place.
Okay. So my next question, why are 4 nucleotide anticodon awkward?
Now you're asking really hard questions. Okay. So anyway, if you try to expand the anticodon from 3 nucleotides to 4 nucleotides, you can't do that in this loop because you have this heavy modification of this base. So that limits the frame to 3 nucleotides. You can add another base. So when people try to do this synthetically, they add another base and then they try to develop some means for recognizing a 4 nucleotide anticodon. But it's very awkward. I mean, it's very awkward to evolve a system with a 4 nucleotide code because basically you're set up with a system where there's so much ambiguity in coding that it's not a system that could potentially evolve. So there's a reason why we ended up with 2 Watson-Crick positions and 1 wobble position. And we were never able to add more amino acids by locking down this position. That basically, if you try to lock down this position at this point, that would become a lethal mutation. But if you add another base in the loop is no longer tight and no longer stable. So that's one problem. The other problem is to actually get it to code for something. You get a lot of ambiguity of coding with a 4 nucleotide anticodon, so it would never evolve. Mathematically, it's maybe not impossible to consider, but to lock down such a 4 nucleotide anticodon is problematic. I mean, people are trying to engineer these things and they're having some success, but the success is a little bit mixed because it's so hard to get specification with a 4 nucleotide anticodon.
So you mentioned earlier about if the 34 position is locked down, that can lead to lethal phenotype. Can you elaborate a little bit more on that?
Oh, so if you want to, at this point, if you wanted to create a superorganism by locking down this position and adding a bunch of amino acids into the code.
Yeah.
So now we're trying to make a master race, I guess. And the problem with that is if you, that the system is so dependent on wobbling now. If you try to suppress wobbling at this position, you will kill the organism. So this change was done before Luca, so it was done very early, so we could, and there was a cost benefit. So the benefit was that you could make the code bigger. You could expand beyond 8 amino acids. So now we're at a situation where we're locked in at 20 amino acids for the most part. If we try to lock down the 34 position to expand the code, that's a very awkward situation for any organism that's dependent on degeneracy at this position to specify its amino acids.
Thank you, Zach.
Oh, those are hard questions. Okay. Thanks. Okay, so let's stop the share. And are we set to stop recording? Okay.